working in home surface lab. And we'll hear everything about single cell analysis today. Feel free to ask questions if you don't understand anything.
Yes. Okay, so yes, today basically talking about different types of single cell analyses that we have. Second. And then, yeah, so essentially I'll touch on essentially when all these technologies first came on board, how rapidly it's been adapted. Then I'll go into essentially what are the methods that are available to generate these types of data, and then touch on a typical overview of what an analysis looks like with some examples along the way. So yeah, that's the latest topics. Wait a second. Quick pause. I'll just pause while I yeah. Any? Okay. Yeah, sure. I'll take one of those guys. Well, maybe. We'll see how everyone's going. Continue down. Everybody's got their favorite. Yeah, so I'll start off talking about when these things first came available and how rapidly they've grown, and then talk about the different methods that are available for each technique, and then the analyses that are involved in them. Okay, so basically, single cell omics became a thing sometime around 2009 with this Ting et al. Paper that introduced the idea of single cell transcriptome sequencing. And then essentially every year, this is from a review I found, so into 2021, but you can essentially see it's been a pretty rapid evolution of additional technologies, basically adding more reads, more complexity of how you organize so you can mix and match different cells and samples together, and then different types of omics coming on board, single cell Hi-C, so chromatin interactions, methylome sequencing in 2012 and 2013. And then another big milestone time point was in 2015. That's when we first get single cell ATAC-seq, as well as multi-omic analysis. So that could be RNA and ATAC, or sometimes RNA and protein, those sort of things, or RNA and methylation. And then another one big time around that time frame that's important is these droplet-based techniques first became available. And that's what 10x Chromium is derived from, is a droplet-based approach. And then just a year later, then in 2016, this Stahl et al. Group is when the first modern iteration of spatial transcriptomics became available. And then it just keeps on going from there. And so as far as how rapidly these things have been adopted, this is the single cell ATAC-seq. So starting around 2015, the number of publications per year is pretty much of a rapid growth, although still not tens or hundreds of thousands, still just thousands of papers are using single-cell ATAC-seq. So it's not, as you'll see in a second, not nearly as broadly adopted as the other methods. Single-cell RNA-seq now has just blown up. Whereas, yeah, hundreds of thousands of articles are published with some term related to single-cell RNA. All I did was search for the word single-cell RNA-seq or scRNA-seq, that kind of thing. And yeah, it has blown up. And then in spatial, even though it really only starts in 2016 here, it's been more rapidly adopted than the ATAC-seq side of things. And it's still also really growing quickly. Okay, so then I'll touch on how, like, what are these different methods? What do they look like? Which ones are the most like broadly adopted? And then talk about how those things work. So as you can see here, this is just a list that ended in 2018 from a review article of all the different types of single-cell RNA-seq methods you could choose from. And I was trying to get an idea of, well, which one should you choose, based on quality or just based on how broadly adopted it is. So I went and tracked down citations for every single one of these methods, and that's what's plotted here. And I guess the main takeaway being that these three methods are all droplet-based single-cell RNA-seq approaches. And Chromium 10X is, even though it has a little less citations, it's probably the most broadly adopted version, and even more so than the original method itself, which is down over here in this dark purple color. But one of the main differences that I could read from the different approaches between like NDrop and Chromium is that it seems like NDrop, the advantage there is that you can kind of generate that in-house. Like it's not a kit-based approach, so there's pros and cons, but I think it makes it easier for a lab to decide, hey, we're going to buy all the parts ourselves and just generate data using this method.
It doesn't require a specific
From what I can tell,
machine or.
yeah, no.
Okay.
I think it's something you can pretty much just say, I'm going to grab all the parts and start doing it. But when you look at data comparisons, as far as like various quality control metrics or like how many genes you get per cell, Chromium still seems to do kind of better. So there's a reason it's so broadly utilized. And so this is kind of the breakdown of how these different approaches work. So again, these are all the droplet-based methods. NDrop, essentially, you're taking your cells, you have little hydrogels that already have primers and the enzymes for reverse transcription present there. They mix together and they all get encapsulated together within individual droplets. So in theory, you have one cell plus their reaction mix. You do your reverse transcription, break up all the droplets, and now you just amplify linear amplification, and then you're ready to sequence. So these are all somewhat similar. DropSeq introduced the idea of having like microfluidic devices, but still in theory, it's roughly the same concept. You have your cells in suspension, it's mixing together with your lysis buffer and an oil, so you're having an emulsion. And then these microparticles have essentially probes on them that as the cells pretty much immediately lyse in the droplets, you're then hybridizing to the transcripts in the cell so that you can then pull those out and sequence. And then finally, this is 10X approach. This is the GEM that people always see, the gel bead and emulsion terminology. Again, using a microfluidics device. So they have their gel beads that have primers so you know which one it came from. And then it's mixing in the microfluidic device so that you have cells coming in from one side and your reagents, the oil, and you're, in theory, encapsulating a single cell with its necessary reagents in each of those droplets. And then you can break those up to your cDNA amplification and then sequence. Okay, so moving on then to single-cell ATAC-seq. Obviously, there's a lot less. Even though it's been around since 2015, it hasn't proliferated to the same degree as the other ones. And from what I can tell, it really does seem like 10X has the lion's share of the market here, both in publications and just in the cost-benefit analysis of it. The cost per cell is down here at the bottom, just kind of get an idea of how much does this cost a person to run these things. I really didn't see major reasons why you would choose the other ones, other than maybe the S3 ATAC approach. You do get more total unique fragments, but one of the more important metrics of ATAC-seq is seeing how many of those fragments end up in peaks. So those are the regions you're calling as actual open chromatin and represent regulatory elements in the genome. it's really not significantly different than 10x, that is less, but then the cost per cell is way, way, way higher. So from what I can tell, unless you have a very specific reason to use one of the other approaches, you should probably just go with the one that everyone else uses.
Multi-ohm is better.
Yeah, and the multi-ohm, you get a little less from that side, but the advantage there is that you're also getting RNA-seq data, so you're getting transcript information from the exact same cell. So you can do independent single-cell RNA-seq and independent attack, integrate that data if you're pulling from the same tissue type, but you're not getting truly, like literally the exact same cell from each one. In this case, with multi-omic data, you are at a slight loss of information. And the approach 10x uses for single cell attack is roughly the same as single cell RNA-seq. In this case, you're starting with nuclei instead of the whole cell, chromatins in your nucleus. And then that gets encapsulated in those same gel beads and emulsions. And then the TN5 transposase is the enzyme that both fragments and ligates the adapters for sequencing downstream. And of course, just like in single cell RNA-seq, the advantages of these single cell methods over bulk is that the idea that instead of getting an average signal of all the cells in your sample, you're truly understanding what individual cells are doing, what their chromatin, repertoire looks like, so that you can start doing things like, well, I have all these different cells. Can I identify a differentiation trajectory and see not only how does the transcriptome change as cells adopt a particular cell fate, but what regulatory elements are important for those transitions over time. And, yeah, so you can do that kind of thing. And that could be developmental trajectories, differentiation, or delete, or, you could have a situation where you're treating with a drug, and so then you can track cells that did not have the treatment versus did, and then how does that change the chromatin landscape in those cells as well. And so, yeah, a lot of those things you can't really do with bulk unless you have a way to really get pure populations of cells, which for a lot of cell types isn't really feasible. Okay, so then how is spatial transcriptome different? One, this is a figure I found showing basically the number of institutions that have the different technology available. And again, Visium is crushing the field as far as 10x is the one that most institutions have as their primary means of performing this technique. You don't need to look at all the details over here, really. The main, I'll show 10x's approach in a second. But really the idea simply here is that not only are you getting the transcriptomic information of individual cell, but you're retaining where from the tissue that cell came from. So that's really important, particularly if you're looking at like rare cell types. So you can, on some of these techniques, you can use like a laser to pick individual like narrow regions of interest and pull cells directly from that region, if that was what is most interesting to you. With Visium, essentially what they do is have a slide that has positions already known on there, and then you lay your tissue on top, and the spots essentially ligate to transcripts within those individual cells. So now you have the transcript and you know exactly where on that tissue the position was. And then the downstream analysis, I'll get into a little bit more, but essentially you're going to be able to identify which genes are spatially variable as well as variable between populations. And then you can use that to identify particular regions of your tissue that are behaving similarly or interacting with one another. So that's a big advantage here, is you can do a lot of really neat cell-cell interaction type information. So yeah, there's, I was touching on there's like 4 broad categories of spatial transcriptomics methods. That's sequencing-based, probe-based, imaging-based, or this image-guided spatially resolved method. The sequencing-based approach, this is what 10x Visium is. Now the HD is a little different because it uses probes, but similar concept, where, as I was talking about, you have a tissue slide. These little dots are all the individual spots on that slide that have barcodes to ligate to your transcripts from the cells and to be able to resolve where it came from in the position. And so you're generating your library, doing your regular sequencing, and the downstream analysis. Probe-based, here you essentially are picking a set of genes that you're interested in studying. So you're not getting whole genome information here, because you're doing like fluorescent probes and barcoded RNA probes. And then this is what geomics, another popular choice, is as well. And so you're picking individual regions of interest on the tissue and then having your probes there to ligate a specific set of transcripts that you're interested in. I can't remember exactly how many genes, but maybe a few thousand, I think, not the entire like 20,000 genes in the genome. And then they're using UV to cleave those from your tissue site, and you know where they came from, and then doing the same sort of downstream analyses. Imaging based is even more restricted in that because you're using fluorescent probes, you're limited on the number of different fluorophores that are available. But if you maybe had, a handful of genes that you knew from past research in your group or from the field were really relevant to what you want to study, then this might be an approach that you could really narrow your focus on a particular region of the tissue and look, well, what are my handful of transcripts doing exactly in these different cells? And then the image guided one is similar to imaging based, but instead of like hybridizing to the entire tissue area, you're using a photoactivation laser to pinpoint specific cells, then pulling those out and sorting them through like FACS sorting, and then just doing typical single cell RNA-seq on those. So you knew where they came from because you had the original image, but it's not linked together in the same way that the like PCEN approach is, where it's all in one go. This was new to me, that approach, so I'm not sure how popular FunSeq is as a method, but could be relevant if you're really trying to narrow in on just one particular cell type that you want to study. And so yeah, this in a little more detail is basically what the 10x one is. So this is that slide I was representing each of these spots. It started around 100 microns. So to begin with, it wasn't actually single cell. You're getting groups of cells in an area. Now it's with the HD coming on board, it's down to 2 microns. Like they have 2 and 8. They typically you treat it at an 8 micron level, which is roughly single cell. In theory, if you were looking at the 2 micron level, you might even get subcellular resolution. So you might be able to say where in the cell a particular transcript is located, as opposed to just which cell has that transcript. Okay, so those are the methods. So now we'll switch over to what do these analyses look like. So first up, this is basically everything someone would typically do for a single cell RNA-seq experiment. And really the whole top half is really just getting ready to do anything that's of interest downstream. And that can be a lot of effort to get just to that point. And typically any kind of next-gen sequencing experiment, the first thing you're simply doing is doing QC on the actual sequence reads. What's my mapping quality to your genome? Do I have any kind of bias of just GCs or ATs in my sequence reads that would suggest something went wrong sequencing? And then the main thing we're kind of always starting with is, can we generate a count matrix? So we have samples on the y-axis or on the top, and then, or I guess it's X, and then genes on Y, and then how many times do we see that gene transcript show up in that sample? And that's basically where you're starting from for everything downstream. And so the first step, as is kind of universal to all of these approaches, is some type of QC. And that's challenging because QC here, there's no like hard and fast rule for what is good, what is bad. It's always qualitative to some degree, to where you have to make decisions like, a typical filter would be removing cells with a really high mitochondrial DNA content because you think that's basically junk. But if you know your cells of interest have high energy requirements, then you would expect them to have at least a somewhat higher than normal mitochondrial DNA content. So you don't want to drop those out. So you have to create these thresholds of where you make your splits for what's good and bad, using some of your own knowledge on, well, what am I expecting to see? What I do typically for some of the other things, like tracking the number of UMIs that's in a cell, the number of features, so that could be number of genes that have a transcript. To try to be as unbiased as possible, a lot of times what I'll do is simply say, if it's below the 2.5 percentile or above the 97.5 percentile, I'm going to throw those out so that I can hopefully try to not be basically cherry picking my data to some degree. I don't think everyone does that, but that's what I found for myself to be a way I feel like I'm being consistent with how I treat data every time before I'm doing anything downstream. And then the next typical procedure, and we talked about this a little bit in the statistical methods meeting, is basically normalizing that. And you normalize not for differential expression analysis where you would still use the raw counts. You normalize for doing dimensionality reduction, clustering, and for a lot of the visualization procedures so that you can look on a plot in a fair manner to say, does this look higher or lower than the other one? And the one approach we talked about a couple weeks ago. It's typically, you take the log of these counts plus 1. That way, if you have a 0, you don't have an issue with taking the log of 0. And then you scale that by some factor, typically 10,000, and that gives you your normalized count matrix. From that, you can identify which genes are the most variable. So this HB is just highly variable genes, form dimensionality reduction, like PCA, component analysis, cluster. And then now that you have different clusters, you can identify which of those highly variable genes are basically uniquely defining each of those clusters and try to use that to annotate your cells. And really, now you actually have something you can ask questions on and explore in more detail. And a lot of these methods are exploratory in nature, in that you're just mining the data to see, well, what is different, to essentially allow you to then create an additional hypothesis of, oh, this gene is really elevated in this cell type. Maybe it's important for that fate determination. So now I'm going to go back to the bench and see, can I knock that gene out in a study environment? So yeah, a lot of this is exploratory. And yeah, you do things like differential expression analysis. So you can compare one cluster to another, what makes them different from the transcript level. You can do the gene set enrichment analysis we talked about, checking gene ontology terms and saying in this population, which ones are elevated or are under enriched. And then you can take gene set enrichment analysis and then do gene set variation analysis, where now instead of comparing genes between populations, you're comparing entire gene sets. So, is the primary RAS pathway elevated in this population versus another one, as opposed to just looking at renin versus one or the other. And then you can also do motif enrichment, so identifying transcription factors that might be relevant to those cell types. One of the challenges there is that the motif that you're using to identify a transcription factor is generally the same for all members of the transcription factor family. So you don't really know specifically which member of the family, because there could be 3, 4, 5, sometimes 20 different members that make up one family. So one way that I've seen and I do to kind of get at that information is just simply looking, well, which one of those members is the most highly expressed in that cell? And then, as a proxy saying, well, that would be maybe the most likely one to be relevant here because it's the only one that's expressed. So I would be, less likely to think it would be one of the other ones. Now, of course, RNA expression doesn't always translate to protein abundance. So a protein could be very present, but only a few transcripts was sufficient to keep it there. So it's not foolproof, but that's basically one way to do it. And then you can also do things like establishing a trajectory. So you can essentially pick, I'm starting here with this cell population based on prior knowledge, and I'm ending over here, and let the algorithm try to decide which populations of cells am I going to go through to go from that starting point to the end point. And so then that can be really fascinating, because you can see through either time, whether it's developmental time or differentiation, what's changing along that trajectory. You can see how different cells interact with other cell populations. So each one of these circles is a different population of cells in a data set. And the lines are basically showing the number or strength of interactions between those cell groups. You can just basically catalog which cell cycle genes are active, and you can categorize your cells based on where they are in the cell cycle. And then spatial transcriptome is really just kind of a subset in the end of single cell RNA-seq, because you're still ending up with your transcript. And then the only addition now is that you also know where those populations of cells came from. But I'll talk more about that in a moment. So here's some examples of what this might look like in practice. So this is actually doing single cell RNA-seq on a cell line. So, we treated cells with this drug that inhibits protein kinase A, and we can show, as we expected and kind of knew, that renin expression is sufficiently significantly decreased. And then so here we're visualizing all the single cells. So here we're not labeling populations of cells, but basically all the same cell type, but have different gene signatures. So something about this treatment essentially is pushing cells down a path to change how they are basically being regulated at the transcriptome level. And then we're trying to see, are some of these like essentially a stable endpoint after that? And do they have features that are interesting to us that we want to study more? And so one way to look at that is essentially just looking at with treatment, what is the proportion of cells that end up in these different gene signatures? And we can say like, oh yeah, this population has really low renin expression, the thing that we were most interested in studying after this drug treatment, is significantly enriched after treatment. And so maybe we want to look closer at that population of that subpopulation of cells to see, well, what's different about it that might be relevant to the regulation of renin. And then, so yeah, here's some examples of doing gene set enrichment analysis. This is at looking at the entire population of cells, just compared the treatment to the control. And then this is looking at that subpopulation where we have really low renin expression and doing the same sort of thing. And we can see like, oh, interesting enough, the epithelial mesenchymal transition is enriched in these cells. P53 is enriched. And then conversely, we're losing MYC and mTOR signaling on the backside. And then this EVA statistic is basically looking at the variation of expression among all these gene sets between the data sets. And basically it looks like when you treat with this cell, you're essentially restricting the number of pathways that are active in the cell. So you're really simplifying what's happening. Yeah.
I have a good question. In this plot, the C.
Yeah.
So what you're saying is 9% of H89 cells have.
We're that rim one low, yeah.
My question is like, okay, when you're treating in petri dish, so all the cells are equally exposed to the drug.
Yeah, but there's clearly, yeah, well, that's the advantage of doing the single cell here versus bulk. That signal might have been masked or muddled to some degree, because like when I'm looking at all of it together, there's not as much of a difference as it is when I'm looking at just that one population. So it could be that, you know, the arrows here are a velocity analysis. So that's basically looking at the ratio of spliced and unspliced transcripts in the data, which you obtain from RNA-seq information. And it tries to predict, you know, what is the stable, which way are the cells going towards. And a lot of these cells are moving towards the RIN1 low population to suggest maybe that's like an endpoint. So maybe we're just capturing it. Yeah, maybe we're just capturing the cells while some have been exposed longer or already transitioned from one type to the other. So,
Very nice.
and then here's an example with single cells from tissue. So obviously you can get different populations that you are annotating based on marker genes. And then you can see interesting things like, oh, the main reason these two groups of populations split is because there's two different lineage defining transcription factors. OSR1 is this whole population, and then FOXD1, which is the renin lineage, are all over here. So if we were really interested in the renin lineage, which we were in this case, essentially we don't care about all these. These are essentially were carryovers that came from isolating the cells, and it's not a perfect procedure. Even with VAX, when you have a fluorescent tag, you're not getting pure populations every time. And then you can look and see, well, of a transcription factor that I'm interested in, what's the distribution of expressions of these are violin plots. You can then here identify, I'm going to compare one population to another and see which transcription factors are likely the ones that are defining that group and making that cell become that cell type. And then you can also use those violin plots if you have marker genes where you're trying to annotate and say, well, I'm really interested in my renin producing cells. Which population is that? So if prior to this being labeled as renin cells, we wouldn't necessarily have known that, but we could look and see, well, these are this is the population of cells that has really high renin and really high AKR1B7, the secondary marker to JG cells. And so that's our population. Then we can compare that to like stromal cells and see, well, which transcription factors are the ones that differentiate this to the other. And we can see like, oh, look, this MEF2 family is maybe really important for fate determination in JG cells. So like this would be the exploratory part where, oh, maybe now we want to go back to the lab and see, can we do cell lines or mouse models where we knock out this transcription factor and see what happens to the population of renin cells. Okay, so that will move us into single cell attack-seq, and then it'll tie back into that example in a minute. So this is less well developed as far as from the analytical side in single cell RNA-seq, and just you saw that it's just not as broadly adopted as a method. So my hands-on experience is predominantly using Archer, and this is from their papers. And of course, they have all the check marks. But there's also Signac and Surratt, which are becoming more popular for this approach, primarily because, unfortunately, it looks like the Archer people, the guy who put that together, his lab, is basically just him. And so I saw he's got a call out for someone else to come on board to try and help maintain this package, which I hope you find someone because it's a really comprehensive, really well-designed package. But right now it's been kind of stagnant for like a year. And so if these things keep going forward, I've started not being able to use it because there's other things I want to do and they're just behind the curve essentially. So hopefully that comes on board because it is really good. You can do a lot of stuff with that. You can see a lot of these other tools. Essentially, the only thing they all do together is take your data, cluster it, and give you visualization.
That is the only one that allows you to do footprinting.
Yeah. Otherwise, to do all these things, you're writing the software yourself to do it.
Oh, okay.
Which, I mean, I guess anyone could do, but obviously that's an entire different time sink of I have to develop a method and then apply it to my data. And method development is a whole other field by itself. So that would be, a whole effort for a paper before you even get to the point of actually able to use that for your own analysis. So it's nice when these tools exist, so you can get to the interesting stuff that prompts more experiments. Okay, but the workflow is not substantially different than single-cell RNA-seq. It's just the terminology or the output of what you have is a little different. Instead of genes, we have features or regions of the genome, but still you're sequencing, you're getting rid of your adapter sequence, you're filtering on some QC on just the sequence data itself and aligning. And then you have some different QC metrics for ATAC-seq instead of single-cell RNA-seq. In this case, the FRIP, the FRIP score, is the fraction of reads in peaks. So you want that to be high. Again, it's a qualitative measure. There's no like I think at one point it was like, oh, if it's below 0.3, you should throw it away. But I've seen data that's way less than that, and it's still really good data. But if you saw something that was incredibly low, essentially what you're seeing then is just like a random sporadic number of fragments all across the genome. So it's kind of a meaningless information. So you might throw that cell out. And then the TSS enrichment score is basically looking at read fragments pile up at the transcription start site of genes. And so you want a lot of your fragments to be around transcription start sites because that's a regulatory element. So that is relevant to chromatin. And so again, arbitrary cutoff with bulk ATAC-seq was like around 6, and then higher is better. But again, you have to just do it kind of based on your own data and somewhat of an intuitive feel of it. If you've seen enough samples, you get an idea of what looks like a passable value. And then the fragment distribution size, the peaks actually represent nucleosomes. So over here is nucleosome-free fragments. The first peak is a single nucleosome. The second peak is dinucleosomes. And if you actually expand it out, you can see 3, 4, 5, 6 nucleosome fragments where they're coming from. And generally, you just, in my experience, this is kind of like looks good, and you're moving on. There's no number here value that you're saying, like, ooh, it's below a certain value, I got to get rid of that sample. So the utility of this feels more like it's only bad if you don't see the peaks. Yes. And then, so here you're still ending up similar to single-cell. You want a matrix that is your samples, your regions. So now the rows instead of genes are regions of the genome. And instead of doing principal component analysis for dimensionality reduction, you tend to use LSI, which is latent semantic indexing. This is a method actually from natural language processing that's been adapted over to genomic data. But you still end up clustering cells. And then a little more challenging part with pure single-cell ATAC-seq data is annotating those cells because you don't have gene expression. But because you have the accessibility information, you can generate a proxy gene expression score called a gene score. So essentially, you're looking at the promoter region and reads that are within the gene itself, and essentially summing that through like a cumulative distribution function to say, this is my predicted expression of the gene based on how accessible it is, and does a surprisingly good job. And I'll show an example of how similar they look with data that was from single-cell RNA-seq and single-cell ATAC-seq. And then you're visualizing and annotating those cells and doing all the downstream stuff, similar to what you'd be doing in single-cell RNA-seq. And so here's some of that, what that is. You call your peaks. So you have individual single-cell signals that you can accumulate and get an aggregated signal. You're good. And then, so each of these peaks represents a region of the genome that is accessible. So the lines, the individual blocks down here are saying this is a region of accessible chromatin, which likely represents a regulatory element, an enhancer, promoter, that sort of thing. And then instead of differential gene expression, you're looking at differential accessibility of a region to compare one cell to another to say these regions are important in this cell and maybe not important in this other one. And then you can also do what's defined as linkage analysis. So you're linking either peaks or peaks to genes, which is you're simply saying that in a particular cell type, whenever I see this peak open, I also see this one open. Or whenever I see this peak open, I see high expression of this gene. So it's basically pointing that this regulatory region, whether promoter or enhancer, is maybe acting on that gene. So it'd be a way to identify the specific promoters or enhancers that are relevant to a gene of interest. And then like single cell RNA-seq, you can do motif enrichment. You can also do footprinting, where you can get an idea of which factor is binding where in a more detailed manner than just predicting the motif itself being present in a fragment or a transcript. So here are some examples that kind of build off what the single cell RNA-seq was, because it came from the same data. But you can do things like, same with the single cell RNA-seq, you can compare two cell populations to see, like here we're looking at parasites, which are also from the same lineage as the JG cells, that FOXD1 lineage, and looking at early JG cells. So these are cells that appeared by E18, embryonic day 18 during development, because they were the first ones that started having high renin expression and AKRMB7 expression. So these would be transcript factors that are relevant to parasites, and then the inverse, the ones that were relevant already to JG cells. And here's that big MEF2 family that we see. And so we can do footprinting analysis with the attack data. And essentially, the higher it is, the more it's enriched in that cell, that population. And you can see all the JG cells and smooth muscle cells and Zentra cells, so other cells of the renin lineage, all have a really strong footprint for the MEF2 family. And this is where I was saying the gene score versus gene expression actually tracks similarly. So, excuse me, the top row is gene expression, because we did have integrated single cell RNA-seq data to go with the attack-seq. And the bottom one is actually the gene score. So this is the gene expression derived from accessibility readings. And so I think the interesting takeaway is that when you have high gene expression, you do see that the accessibility predicted it to be highly expressed. And so even when you don't have RNA-seq data, you actually can feel relatively confident that my predicted value is pretty plausible. That is what the gene is doing in the cell you're studying. And then below, on the left, looking at two different genes, two different loci. So this is renin, this is AKRMB7, because these are really relevant to JG cells. The top rows are the peaks. So the higher the peak, the more accessible that little spot is. And then the order of the cells basically is what we were defining as our renin trajectory. So starting at E12 during development, all the way to post-natal day 30, these were the cells that made up the trajectory. And the thing you see is that the peak highlighted lines are basically where the promoter is. And we can see that there's a peak getting more and more defined as we go, and additional peaks become available. So more regulatory regions are becoming accessible because we're trying to regulate this gene. And that's what the peak to gene linkages are showing. So these are all links showing how strongly correlated they are. The darker the color, closer to black, so that these regions that I have highlighted in yellow are linked to the promoter at renin. And then we looked and saw, oh, there's a motif for the MEF2 family, for the NFI family that we looked at, and some other ones that suggest, well, this is a region that we always see open in renin cells. It's also always open whenever the promoter is accessible, and we see binding sites for these factors that we saw to be enriched when we compare that population to the other ones. So it really kind of just ties the whole story together and gives you a lot of motivation then to want to study those genes in a more detailed an experiment. But that gets back to why on some level this is exploratory, because we haven't confirmed that gene is relevant, because we'd have to do more experiments to do that. Okay. So switch it up to the final topic of spatial transcriptomics. This again is like kind of the general overview. The endpoint for even the image-based or sequencing-based is similar to what we had before, the expression matrix, but now we also have the paired location matrix. So we also remember where on this tissue slice a cell came from. And then you're still doing the same things. There's some tools that are available. The one highlighted is also what I happen to use. I can't remember which paper I got this from, but yeah, I've also used ScanPy, that's Python based, but really most of the techniques are either using Python or R for the analysis. There may be a few that are like graphical interfaces and web-based for people to do to make it a little bit easier if someone doesn't have any kind of computational background, but most of it's done on the command line. But yeah, you're doing things like dimensionality reduction and clustering, just like in single cell RNA-seq data. But now you can identify which genes are spatially variable, not just variable between populations in a dimensionality reduced visualization. You can do that cell-cell interaction and trajectory analysis, that sort of thing. Okay, so here are example results from that. In this case, this is thanks to Monica. First, we had kidney slices from mice where you're fed a normal diet or a diet that was low in salt with the ACE inhibitor captopril. So we expect the captopril treated mice with the low salt diet to have a whole lot more renin being expressed. And that's, they say, this phenomenon of recruitment, or essentially cells of the renin lineage transform back into a renin phenotype. That's what we're really interested in studying. And so you can see immediately the left slice here is the basal normal diet condition. And so you can kind of make out, there's little circles on there. Those are the spots from the Visium array. This is standard Visium. And then the slice itself is from the H&E staining that you can basically overlay and see where on the slice I'm seeing sample. And you can see, like, here's a little glomeruli with some high renin expression right there, that little red dot. But obviously, as soon as you look at the one where it's been treated, there's a whole bunch more renin being expressed. And so that's why we want to look at in more detail. What about these cells differentiates them from their original form? And so we can do analysis of looking at the interactions between cells, the communication that they're doing. And the first takeaway is that yellow is the normal diet, blue line, the blue bars here are when you've induced homeostatic threat. There's a whole lot more interactions, and those interactions are stronger. And then we can say, okay, well, what makes our putative juxtaglomerular or renin cells over here different than the ones in the control slice? And we can see, oh, well, it looks like a handful of pathways are what predominantly differentiate them. Collagens, laminins, thrombospondins. These are the pathways that are elevated in our recruited cells as opposed to the other ones. So maybe these pathways are relevant to that transformation process. And then you can also look and say, so again, this is kind of like what I showed earlier. Each circle is a different cell population that we identified on the tissue slice. And then every line is showing the interaction strengths going on between those two populations. And if it's red, that means we saw increased signals under the homeostatic threat. Blue means it actually went down, but you can see very few have reduced signaling in this context. Most had dramatic increases in signaling. And so we wanted to figure out of those populations of cells, which ones perhaps contributed to a greater degree to that increased signaling. And so the plot on the left, this is all the populations under the normal diet. In this bright yellow is the JG cells. You can see their combined strength of interactions is like 7. And then interesting though, over here, everything, some of these have shifted up and to the right, meaning that the interaction strength is increased. And so JG cells are now one of the higher populations. And then in really in particular, the interstitial cells too, really moved up, suggesting that maybe these cells, podocytes to a degree, and mesangial cells also are contributing to this increased cellular communication that's going on. So suggesting then that maybe these pathways that we identified are relevant between these various populations to induce the change. And then the bottom plots are just highlighting the bar here is JG cells under normal diet. And then over here are those cells under the homeostatic threat. Takeaway being that the total number of interactions has gone up dramatically. The darker the red bar, the greater the strength of interactions are within these pathways. And so there's those collagens and SP1, lemons, VEGF, that sort of thing. So it really seems like these cell pathways are really important to the differentiation or transformation of cells back to that renin phenotype. And so, yeah, that's all I have. So
Yes.
I'm happy to answer questions if I can. Okay. Anybody has questions on Zoom, welcome to ask as well.
Did have a question. On the last slide that